Welcome to the debate. Today we're delving into the world of aerogels, not quite the brittle traditional silica kind that maybe comes to mind first, but actually a sophisticated new generation. These are derived from natural biodegradable polymers like alginate and cellulose. They're incredibly porous materials, structurally really fascinating, and they're showing, well, immense potential in areas like drug delivery and even tissue repair.
That structural capability is indeed pretty staggering. I mean, we're talking about materials that can offer just about the highest internal surface areas imaginable. Traditional silica aerogels, as you said, can hit what, over 1,000 square meters per single gram, which is it's basically the surface area of a tennis court packed inside a sugar cube, roughly speaking. And yeah, this makes them phenomenal platforms, potentially, for carrying and controlling the release of active pharmaceutical compounds.
Right, exactly. But the central question we really need to grapple with today, though, is this crucial trade-off. Do the highly specialized performance advantages that these polysaccharide aerogels offer, and I'm thinking specifically about enhanced drug bioavailability and complex tunable release kinetics, do they actually justify the inherent material instability we sometimes see and the absolute reliance on high technology synthesis processes like supercritical fluid drying?
Hmm.
I believe that the clinical payoff here, the ability to solve these huge, really fundamental drug solubility problems, I think it means the synthesis complexity is, well, it's a necessary price of admission. It validates the entire process, in my view. Superior therapeutic outcomes necessitate this level of precision.
Okay, and I think I'll be focusing on the opposite side of that coin, that the significant practical, technological, and let's not forget economic hurdles really restrict the viability of these materials. They might be a scientific marvel in the lab, absolutely, but they remain, for now at least, a bit of a production nightmare that industry just can't easily adopt on a wide scale.
Well, let me lay out my case then. Aerogels are, I think, uniquely positioned to overcome what I see as maybe the biggest pharmaceutical roadblock today: poorly water-soluble drugs. We're talking about BCS Class 2 or even Class 4 drugs. These are the ones that are notoriously hard for the body to absorb, simply because they just won't dissolve well in bodily fluids.
Okay. And the key is that internal structure, specifically the high volume of mesopores, those tiny channels between, say, 2 and 50 nanometers. This structure, it stabilizes the drug molecules in a non-crystalline state, or an amorphous state. And this state is just far more soluble. We have pretty hard evidence showing enhanced dissolution rates can be 4 to 5 times higher than the crystalline form.
Right.
And if you look at some highly challenging molecules like paclitaxel, for instance, when loaded into a silica aerogel, well, studies revealed a 13-fold increase in oral bioavailability in vivo. That's compared to a standard marketed intravenous formulation. That is a massive clinical leap forward, you have to agree. And to achieve that state, that specific structure, the specialized drying is, well, it's unavoidable. We absolutely must use supercritical carbon dioxide, or SCO2.
That high pressure necessity, though, that's really the crux of the problem, isn't it? Because while CO2 might be chemically perfect for the job, gentle, no residues, that absolute dependence on it immediately raises massive red flags for scale up. You need expensive, specialized high-pressure vessels. You need operators trained in high-pressure systems. It moves production far, far outside the realm of, common pharmaceutical manufacturing lines and equipment. It's a big ask.
Mm.
And beyond just the processing cost and complexity, the base material itself often shows some pretty critical instability. You mentioned alginates and cellulose. Well, many of these natural biopolymer aerogels, unless they are heavily cross-linked or otherwise chemically treated, they just swell up or completely collapse the moment they hit aqueous body fluids. And doing so destroys the entire advantage of that beautiful, open porous structure you just spent so much money and effort creating with that CSCO2.
You focus heavily on the cost and complexity of SCO2. I hear that. But we rely on this seemingly exotic processing precisely because the alternative doesn't work. If we just try to air dry the wet gel, the capillary forces, essentially the surface tension of the liquid evaporating, will violently crush that delicate porous network. It's unavoidable. SCO2 acts as a solvent under high pressure that, crucially, when the pressure is released, it bypasses the liquid phase entirely, turning straight into a gas. And that preserves the structure that is absolutely necessary for achieving things like that 13-fold bioavailability boost I mentioned.
That 13-fold increase for small molecules like paclitaxel is a genuinely compelling number. I grant you that. It is impressive. But it also highlights, for me, the core limitation. You're talking about small molecules, ones that generally adhere to Lipinski's rule of 5, the standard drug-like properties. What happens when we look at the next generation of therapeutics, biologics, peptides, proteins?
Well, the research is ongoing there, but
Because the predominance of those small mesopores, the 2 to 50 nanometer range, it means conventional aerogels suffer from severe size exclusion effects. They physically hinder the uptake and loading of bulky therapeutic macromolecules, things like insulin or complex proteins. They just can't get in effectively. Furthermore, the kinetics are frankly terrible. Achieving adsorption equilibrium via SCO2 impregnation is often a major rate-limiting step. It frequently demands many hours, sometimes a full 10 to 24 hours, just to load the active agent onto the carrier. That kind of duration is highly problematic for throughput and, any kind of modern continuous manufacturing target.
That measured processing time, however, I'd argue is a worthwhile investment when we consider the resultant capability. We get tunable and targeted release profiles. That's precision delivery that is simply impossible with simpler conventional carriers like, say, standard tablets or capsules. The complexity yields an unparalleled level of sophistication in drug release. For instance, we can achieve true gastric protection. We can use techniques like applying an enteric coating onto silica aerogels, maybe using a polymer like Udragite L. This can prevent drug release entirely in the acidic gastric fluid, protecting the drug or the stomach, only to trigger complete, fast release within minutes once the particle hits the neutral pH of the intestinal fluid. That's smart delivery.
Wait a minute, though. I'm sorry. But I just don't buy that these complex, multi-layered coating strategies are really a practical, viable, long-term solution. The very need for such a protection layer demonstrates the fundamental material weakness, doesn't it? In many published works, researchers find they have to pre-coat the silica aerogels, maybe with molten PEG or something similar, before they can even introduce them to an aqueous Udragite suspension for the actual functional coating.
That's a processing step, yes, but
But if the core aerogel matrix itself cannot even withstand a simple, common aqueous processing step without collapsing, its long-term stability when exposed to the much more complex biological environment, saliva, gastric juices, blood, interstitial fluid, that stability remains highly questionable in my mind. You are, in effect, solving an inherent material weakness by just adding more layers of process complexity, which is exactly the opposite direction industry needs to go for streamlined, cost-effective production.
But we are also developing methods that integrate functionality directly into the matrix itself, moving beyond just simple external protection layers. We have successful examples now of stimuli-responsive aerogels. Think about alginate derivatives that incorporate temperature-dependent blocks, like PNIPAM, co-NHAM. These materials can actively shrink or swell in response to body temperature changes, for example, to regulate drug release, creating a sort of on-demand delivery system.
Okay, stimuli responsive is interesting, but does it solve the core fragility?
Well, we are also solving the structural fragility issue, or perhaps bypassing it, through advanced geometry control. Think about pulmonary delivery, right? Where particle size is absolutely critical for reaching the deep lungs effectively. Techniques like inject printing combined with SCO2 drying have successfully produced incredibly uniform alginate beads precisely sized at, say, 4.0 microns in aerodynamic diameter. That's ideal for targeted delivery of inhaled drugs like salbutamol sulfate. That level of geometric control is vital for delivery efficiency, and you can really only achieve that kind of precision via these complex controlled methods.
That level of control is also tremendously difficult to reproduce consistently, especially at scale. And that brings us right back to the viability question. The final properties of those fine particles are intensely sensitive to process variables. Achieving that beautiful, uniform particle size distribution requires agonizingly precise control over factors like the polymer mixing order, the specific type and concentration of stabilizer or surfactant used. You mentioned span 80 versus span 85. In some systems, that makes a difference. Even slight variations in these parameters can result in inconsistent product batches, which is, as you know, a massive quality control headache, potentially a showstopper for any pharmaceutical product. And this sensitivity leads directly back to questioning the long-term industrial feasibility. If we start with large monolithic aerogels and try the simple route of just milling them down, well, we produce irregular, jagged particles. That hurts flowability, which is a major drawback for feeding conventional high-speed tableting or capsule filling machinery. But if we go your preferred route and attempt to manufacture uniform microspheres directly, that requires these complex, often continuous processes like emulsion gelation combined with SCO2 extraction. And that demands strict, real-time, instantaneous control over things like mixer geometry, rotation speed, flow rates. This is a highly specialized chemical engineering challenge that far, far exceeds standard pharmaceutical production capabilities that exist today.
I certainly acknowledge the geometric control challenges, that's fair. But I would counter that ongoing process optimization is demonstrating strong potential for industrial viability, especially when you factor in the efficiency gains often achieved with increased production volume. We are seeing simulation data now for industrial-scale alginate aerogel production, for example, that suggests really significant reductions in environmental impact, things like up to 96% reduction for ecosystem quality metrics, compared to bench-scale production. Efficiency, it seems, dramatically improves as volume increases, making the economics and the ecological footprint potentially far more sound over time.
Simulations are one thing, actual scale production is another.
True, but let's also not dismiss the structural integrity point you criticized earlier too quickly. We have seen, for instance, non-fixated polyurea cross-linked silica implants that demonstrated absolutely no displacement in preclinical models over a 20-month period. Their unique, very low density is actually proving to be an advantage for long-term implantable drug delivery systems, reducing mechanical irritation and the risk of the implant shifting. And the high degree of tunability available via surface functionalization, which is part of this complexity, allows us to potentially perfectly match the adsorption affinity of the aerogel skeleton to the specific drug being delivered. So this complexity becomes a customizable feature, not necessarily just an unnecessary burden.
Okay.
So I think the key takeaway here is fundamental. Aerogels, particularly these newer polysaccharide ones, provide solutions like stabilizing amorphous drugs for better absorption and enabling precise, multi-stimuli controlled release solutions that simpler, perhaps cheaper, technologies simply cannot achieve right now. The inherent complexity of the manufacturing process, particularly that reliance on SCO2, is, in my view, a necessary cost for achieving these potentially unparalleled therapeutic benefits and dramatically improving outcomes, especially for difficult-to-treat diseases or difficult-to-deliver drugs.
And conversely, from my perspective, the material science gap, combined with the process engineering challenge, remains just too wide for broad application right now, until fundamental material instability issues, like that crippling collapse in aqueous environments, are solved more elegantly, perhaps without requiring multiple complex layering and tricky processing steps. These polysaccharide aerogels will likely remain highly specialized, perhaps confined to niche applications. The significant burden of technological overhead, the capital investment, and the stringent quality control needed to ensure uniform product consistency means they will struggle, I think, to move past niche uses into widespread high-volume pharmaceutical production in the near future.
Well, I think our discussion today certainly underscores that fully understanding and ultimately mastering the precise relationship between all these synthesis parameters and the final material properties is absolutely crucial. Whether you prioritize the potentially massive performance enhancements, as I tend to, or you focus more on the immediate challenges of manufacturing scalability, as you've highlighted, that understanding is the key step needed to determine the ultimate role these truly advanced materials will play in the future of biomedicine.
Agreed.
The potential is there, undeniably, but the path to realizing it widely is still quite complex.